Okay. Welcome everybody. Happy New Year to all. We're back with the EBD seminars. Seems that the flu has arrived to the EBD, but anyway, most probably there will be in YouTube watching it. Today we have Rubén, which is originally from Granada, where he did his PhD in genetics. Then he worked in transcriptomics with Locust. But then he went to Illinois, to the States, for 5 years, I think. 4, okay. Then there he did transcriptomics again, but like this time with bumblebees, and he's today now here in Conchita's lab, where he's doing epigenomics and transcriptomics now in different biological system. Now is with plants, and he's working mainly with Erodiums. And yeah, that's all from my side. The floor is yours, Rubén.
All right, thank you. Well, thanks for letting me talk about the recent outcomes of the project that I'm involved with Conchita's lab. So the title of my talk is Exploring Epigenetic Variation in Non-Model Plants, a Genomic Approach. I'm going to start with an introduction on epigenetics in plants and their implications in different aspects of biology. Then I'm going to start talking about my involvement in 2 research lines. The first one is epigenetic variation in plant populations, and the second one is DNA methylation in plant interactions. And I'm going to do a final presentation of the future steps of the research. So I'll start by mentioning that phenotypic variation is important. It's a key factor in natural selection, and also it makes a species have more possibility to confront challenging environments or changing environments. Okay. So one can wonder, where does a phenotypic variation come from? What are the sources of phenotypic variation? So, so there are mainly 3 sources of that contribute to phenotypic variation. We have environmental variation, genetic variation, but I'm going to focus in epigenetic variation, which is compared to the other 2 sources a little bit understudied. Okay. So I'm going to work with the definition of epigenetics as phenotypic changes that are independent of genetic changes or mutations, okay? And there are described mainly 3 molecular mechanisms for epigenetic regulation, okay? We have microRNAs that can eventually have a role in gene expression. We have stop modifications that can contribute also to differences in gene expression, but also in chromatin modeling. But I'll be focusing on DNA methylation, which is my interest right now. So how does DNA methylation works? It is pretty simple. We have cytosines, okay, in the genomic DNA. And DNA methyltransferases recognize these cytosines and add a methyl group. Okay, these can have implications in gene expression, like repressing the expression of a gene or transposable elements. It is a reversible process. There are demethylations that can just eliminate or remove that group from the cytosine. It is also dependent on the sequence. That means there are different methylation contexts. Depends on the position of the cytosine. If it is followed, for example, for a guanine, that's a methylation context that is common in most living in most eukaryotes. Okay. However, in plants we have another 2 additional methylation contexts, CHG and CHH. The H is any nucleotide but but G. Curiously, they are their methylation and maintenance and de novo methylation is controlled by different DNA methyltransferases in plants. All right, I have to say that CHG and CHH are more, these methyltransferases are more involved, are more like susceptible to environmental changes. However, CG context seems to be more heritable and more involved in developmental epigenetic marks, okay? So, yeah. So DNA methylation can contribute to gene expression and then contribute to phenotypic variation. For example, in Linaria vulgaris, we have this flower type and morphology with bilateral symmetry, all right? And apparently this is due to a gene that is involved in flower morphology, that is L-STIC, that when it stays unmethylated, the promoters are unmethylated, we got this wild type, let's say wild type flower morphology. However, when methylation is present in the flanking regions of this gene, the expression of the gene is inhibited, and we can see that this radial pentamer flower type is shown instead. So this is a contribution to a phenotype variation. Okay, so DNA methylation also, since it is dependent of the sequence, the DNA sequence, you need literally a cytosine in methylation context to have it or not to have it. Sometimes we can try to detect if there is epigenetic population structure in natural populations. For example, in 3 species of this Dactylorhiza orchid, there were different localities that are shown with different shapes. The species are the different colors. We can see that some species in different localities, they have almost no epigenetic structure. This is DNA methylation divergence. Okay. However, some species, even in the same population, they have quite the epigenetic divergence. So this is interesting because in wild populations we are not very aware of how common is this or how uncommon. In addition, DNA methylation can be also responsive to plant interactions, like for example stress. For example, in tobacco, when tobacco plants are infected by the tobacco mosaic virus, we can see that the gene helix, which is a gene that is involved in the pathogen response in plants, showed shifts on its methylation patterns in the DNA sequence in less than 24 hours, and they were maintained at least for another 24 hours. We can see in the uninfected and infected plants when we cut, when we digest the locus with restriction enzyme that is sensitive to methylation, we can see that the patterns are different. And just to add for more evidence of DNA methylation and phenotypic variation, tobacco plants that were knocked down by the methyltransferase produce this dwarf phenotype. So with all this, I want to show you the results that we are having, or the experiments we are doing in 2 different research lines. The first one is it tries to tackle understanding epigenetic variation in natural plants populations, and the second one will be understanding epigenetic regulation in plant interactions. So, as we say, we can detect sometimes epigenetic structure in natural populations. So I wonder how extended is that. So I'll be flocking around these 3 questions. Is epigenetic variation a structure in natural populations? Is it common? Is it uncommon? Is it true? Is it false? Does epigenetic population structure differ between congeneric species, but that have like different lifestyles or life histories? And eventually, since I got the tip in the title, since this is a genomic approach, I'm going to be interested in which genomic regions, genomic elements are differentially methylated between the population or species. Note that when I say epigenetic here, I'm going to refer to DNA methylation mechanism, okay? So, the species system I'm going to work is it consists of these two species of the genus Erodium, which is Geraniaceae. We got Erodium cazorlanum, which is a perennial plant. It has, it's woody, and it's an endemist of Sierra de Cazorla. Okay. In addition, it grows only in dolomitic soils, and also it's an I think it's an octoploid. So its genome size, it's quite big compared to other. In contrast, we have Erodium cicutarium, which is an animal herb. It is a herb, herbaceous, it's not woody, and it is widespread. We can find it in almost all the Mediterranean basin. It's even an invasive species in the USA, so it has resources. It likes soils that are like more developed. It's supposed to be diploid, but some individuals seems to be tetraploid. So the sampling size is this, Sierra de Cazorla. We were sampling for 6 populations of 3 of Erodium cazorlanum and 3 of Erodium cicutarium. I got them marked here. I have to say that despite there are some overlaps between the population, the location names, since Erodium cazorlanum grows in dolomitic soils and Erodium cicutarium grows in more developed soils, they were never like growing together. So we can consider their different populations despite sharing the name of the locality, okay? So what we did was sampling 6 individuals per population, so we ended up with 18 individuals in for cazorlanum and other 18 for cicutarium. We sampled leaves. We extracted DNA from these leaves, and with each sample we divided the aliquot of DNA into 2. One of the aliquots was used to generate rad sequencing to estimate the genetic structure of the population. At least something that could guide us is this population already has a genetic structure or not. So we aligned the reads to the Erodium cicutarium draft genome that our lab has produced. Then we perform a SNP calling with stacks. After the filtering of these SNPs, we ran a mixture analysis, and eventually we managed to get a list of SNPs and also the data that allow us to screen the genetic structure or not. So we have the other half of the DNA for all the samples. What we did was treating it with bisulfite. I'm going to explain why I'm doing this. So bisulfite conversion, it is key for the study of DNA methylation, because we have cytosins that are methylated and unmethylated cytosins, correct? So once bisulfite is treated to the DNA, cytosins that are unmethylated turns into uracils, while methylated cytosins are protected by for the effect of bisulfite and remain a cytosine. Methylated, but it's still a cytosine. After several cycles of PCR, uracil, since this is DNA, it is turned into thymine. So eventually, if I have a reference, I can align the reads treated with bisulfite with the reference genome, And if I have a match with the cis, that's because it's going to be a methylated cytosine. And if it got a T when a C should be, that's a methylation position. And I can then estimate the percentage of methylation in that position. All right. So once the DNA was treated with bisulfite, we made the bisulfite RAD-seq analogous, and then we aligned these reads to the cicutarium genome using Bismarck, which is an aligner that is sensitive to methylation, DNA methylation detection. Then we checked for differential methylation between populations, separately for both species. That gave us a list of DMCs, which is differentially methylated cytosines. We use these DMCs as markers to estimate epigenetic population structure. And then from those differential methylated cytosines, we see which ones were like close in the genome to generate a list of differentially methylated regions. These differentially methylated regions could be tracked down in the Erodium cicutarium genome, and we can see whether it is overlapping a gene, an exon, an intron, a flanking region for a gene, an intergenic region, transposable elements, so we can genomically characterize them. Okay? So the genetic the population genetics of both species, we end up with this number of SNPs, more than 20,000, more than 30,000 in Cazorla and cicutarium, respectively. So the PCA in Cazorlanum is tricky because we can see that there's some stratification there, where the variance was not, the explained variance from the two first PCs was not great. And the admixture analysis revealed that the most valid K for these populations was 1. That means that all of these would be the same population. So it seems that there's not a strong or not at all genetic structure in Erodium Cazorlanum in this population. Where with Erodium cicutarium, we got that two of the localities cluster almost together. They stick together, and the third locality had like 2 clear groups. One of these 4 with these 4 individuals, another with these 2 individuals. The variance explained by the PCA for the first PC was like 20%, combined with this other one, that's 33% of the variance. That is definitely more than what explained in Cazorlanum. And the admixture analysis revealed that the most the most probable outcome is this, that there are like 2 genomic entities. However, it was almost a technical tie with the 3 based on the cover. So you can see that in these 2 populations it's more or less homogeneous, and in this population, Puerto Tejo, the blue one, we have like 2 clear distinct groups. So what happens with epigenetic population structure? Well, first of all, I have to say that we had a little bias between Cazorlanum and cicutarium. We detected more differentially methylated cytosines in Erodium cicutarium compared to Erodium Cazorlanum. This is probably because we are using the reference genome from cicutarium, and probably we have more matches there. But anyways, we can run analysis to have an idea of what we have, and using all methylation contexts, this is CG, CHG, and CHH, we can see that in Erodium Cazorlanum, almost all the samples for all the from all the populations cluster more or less together. There are some individuals that were like skipping that tendency, but there's not a clear epigenetic population structure. In Erodium cicutarium, well, it seems that there's a little bit more separation, but there's still like this main cluster of individuals that are sticking together. The hierarchical clustering of methylation percentages was more or less the same. There was like a limited clustering in both species. So I run this analysis in the three separate contexts. CHH and CHG were a mess, like literally, I didn't see any thing resembling this. However, CPG context was pretty consistent with all methylation context again, sorry. So we can conclude that epigenetic structure is stronger within population than between populations. We have a lot of individuality epigenetically talking. So now I'm interested in the regions of the genome that have different levels of methylation. So I managed to pull together from this set of cytosines that were differentially methylated between populations a number of differentially methylated regions, which is DMRs. So we got more in Erodium cicutarium because we started with more differentially methylated cytosines. But we can see that the number was estimated was higher for the CHH context. The next question was, what are they overlapping with? What are the differential, the elements of the genome that are with more differences in methylation? So for Sierra de Cazorla, we detected that mostly exonic parts are the ones that are with more differences. It happens more or less the same in cicutarium, but it is more like compensated by the CHH context, which is more a wild card. It showed also a lot of overlaps with introns, with intergenic regions, and also 5 and 3 UTRs. The interesting thing that we found about transposable elements is that copia elements were enriched in the set. So that means that compared to the distribution of the transposable element families in the whole genome, we found that copia elements were overrepresented. This could indicate that transposable elements can have a regulatory role, or at least they are coupled to DNA methylation regulation in this species. So just some conclusions about this first line. We found no population structure in Sierra de Cazorla and weak structure in Erodium cicutarium. The epigenetic variation was higher within populations. We managed like to improve a little more this epigenetic structure just focusing on the CPG context, but not that much. And finally, we have the copia elements, transposable elements that could be involved in DNA methylation regulation in Erodium. All right, so I'm going to start with the second line I'm involved. This time it's about epigenetic regulation in plant interactions. So we see, I showed you an example with the tobacco that its methylation pattern in a gene was apparently regulated by DNA methylation when exposed to a stressor. In this case, it was a virus. So this is common in life, that you have your epigenomic marks to generate your wild type phenotype, but suddenly a stress source messes up your methylome, your epigenome, and that can have consequences in the phenotype. The extent of those consequences depends from species to species, and if we have to take as an example the lack of epigenetic variation and the high individual variation, maybe from individual to individual too. So if we manage to manipulate the epigenetic signals and expose those experimental manipulated individuals to experimental stress sources or interaction sources such as herbivory, infections, drought, whatever, we could more or less explain what are the trends or what's happening or how involved is epigenetics in the response to that stressor, okay? So with DNA methylation, we have the luck that this molecule, azacitidine, it's a blocker of DNA methyltransferases. That means it competes with it and makes it not usable. So this means that when we have a fully methylated site in the DNA, when the DNA methyltransferase is not blocked, what happens is that there's after a cycle of replication, we have one of the threads is still methylated and the other is not. This is called hemimethylation, okay? And then the DNA methyltransferase is going to recognize, hey, there's only one methyl group here. I need another one here, okay? So it will add this group. And then after a few cycles of replication, the methylation in this position is maintained. What happens if we block this with azacitidine? Well, there's no enzyme that is doing this. So after a few cycles, this thread is gonna be like overshadowed by the rest of the threads, and eventually these sites are gonna gonna stay unmethylated. All right. It works for DNA methyltransferases that recognize CG context. However, CHG and CHH contexts in plants are recognized by older DNA methyltransferases, and azacitidine doesn't seem to affect them at all. So at least we have this DNA methyltransferase 1 blocker. So this opens the field to try to see how DNA methylation can affect phenotype in plants in experiments that are manipulating stress response. For example, we set an initial experiment, a transcriptomical experiment, just to see the effect per se of azacitidine in Erodium cicutarium, the herb that I used in the other experiment. We were interested in the genetic expression differences between control and azacitidine-treated plants. We also were interested in how it's affecting the phenotype of this plant. And eventually, if there were associations between phenotype and gene expression changes derived from this treatment. So, in order to do so, we got from a single Erodium cicutarium parental, we got seeds that are full siblings, okay? These seeds were planted in a greenhouse. And 12 seeds were just not treated with azacitidine, while another 12 were treated with azacitidine. We let the plants grow for 5 weeks, and then we sampled leaves and roots from those individuals. We phenotyped the leaves and the roots, so we could perform a trait analysis or analysis of the samples. And for a subsample of these 12 individuals, 6 and 6, we took leaf RNA and root RNA and performed RNA-seq and generated RNA-seq libraries to check the gene expression in leaves and in roots. So these reads were aligned again to the Erodium cicutarium genome. Once we got the read counts per gene, we ran differential expression analysis with DSIC2. And also we were interested in see if in the transcriptomical network that we can generate with the correlation of expression of different genes, if there were modules, and if these modules were related to the azacitidine treatment or not. So we calculated this gene expression network with WGCNA, and got some co-expressed gene networks. And then we correlated these modules that were associated with the azacitidine treatment with the phenotypic changes. So the first analysis that I'm going to explain is the differential expression analysis, okay? So the first thing that I noticed is that there's a huge effect on tissue expression. Like you can see the differences in numbers more here than here. This is like azacitidine affected 151 genes. However, the difference between leaf and root were like almost more than 50,000 genes. Okay, so yeah, there were some overlaps. Yeah, okay, and it's not a bad number, 151, but still the effect was really big. What is interesting is that one of the DNA methyltransferases that is not that is associated to methylation in CHH context got upregulated. So maybe there's a compensation upregulated in the azacitidine treatment. So maybe there's a compensation mechanism. So this was interesting. However, I wanted to atomize a little bit and eliminate the tissue effect. So I analyzed differential expression within tissues, and this led to a little bit more azacitidine. We got like 101 in leaf, 417 in roots. So definitely there's more sensitivity to the treatment in roots. When I ran geoterm enrichment analysis to see which tags, which functions, which biological processes were like overrepresented in the sample of those differentially expressed genes, I noticed that a lot of them were related to transposable element transcription. So what happens in plants is transposable elements are usually methylated to avoid their expression. If a transposable element is a retroelement is actively expressed, it can insert in areas on the genome that can provoke genome instability and can mess up with the viability of the plant. So it makes sense that the plant wants those elements silenced. However, under stress or demethylation, these signals are like being removed from the element, and it's provoking them like to be active again and can provoke that. So this can have consequences in the genome stability and eventually viability of the plant. So this happened in roots. So for the co-expression network, I just wanted to show you that we managed to pull these 7 modules of co-expressed genes. There's a huge module that is associated with tissue differences. However, the second module, this blue one here, had a really strong association with azacitidine treatment. We can see that the expression of the genes were mostly upregulated in the root, in the azacitidine treated root, and again the gene ontology terms that were overrepresented in those genes that were belonging into that module were related to transposable elements. So this is the transcriptomic part. The phenotypes. So we phenotype leaves and roots, okay, and we managed like to run multi-dimensional scaling analysis with it, and it led to the 2 dimensions managed to explain root morphology in dimension 1 and leaf morphology in dimension 2. We can see that this part of the multivariate analysis is correlated with roots that are more absorptive but has less volume. However, this part has stronger roots that are more keen to store resources rather than be absorptive. More or less happens the same with leaves. We have like super efficient leaves that are like with a really good photosynthesis rate, but it's not a lot of energy involved in them. However, this part of the axis is explaining resistant leaves that are more concerned on protection rather than in production. Okay, so the green dots are the control individuals. We can see they're concentrated here. Okay, and the red dots, you see them all scattered. That's the effect of azacitidine. So it's literally, it was not so directional, but definitely increased the phenotypic variation. And the effect was stronger in the roots. We can see that there are almost no control individuals in this side of the pie plot. But there's plenty of associated individuals there. So it seems that it has an effect on, at least in root phenotype. So now we are checking these modules that I was talking about on the phenotype, on the traits, the plant traits, and also the dimensions that we produce with the multivariate analysis. Okay, so I was interested in the blue module because it was the one that has like a response to azacitidine, transcriptomically speaking. We see that the expression in leaves was not correlated to any trait. However, in root, in the root expression of this module, we have a strong correlation with the first dimension, which was the one that was defining the root morphology, and also with a single trait, which is a specific root length. I think it's the proportion of fine roots with relation with the mass over biomass, I think. So we have positive correlations here. So it appears that that module, the genes that are in that module, are in a way involved in the root morphology. And it seems that could be also regulated by DNA methylation at some point. So the conclusions of this study is that both gene expression and phenotype show more variation in roots treated with 5-azacitidine in Erodium cicutarium. The genes upregulated by 5-azacitidine were enriched in transposable elements, at least in the roots, and the gene module 2 was associated with 5-azacitidine treatment on root traits. So this is a preliminary study because once we got this, the idea is, I'm now explaining the future steps, is adding an additional level. We got the effect of demethylation. Now we want to see the effect of demethylation and a plant interaction. Like for example, we got already Geraniaceae generated data from an experiment that is involved in herbivory. So it's more or less like the experimental setting that I explained. However, we add a treatment of artificial herbivory, which we just punch the holes in the leaves or cut. The leaves were like cut, like literally doing some gardening. Another level of herbivory was like treating the cut plants with goat saliva to see if there are like different responses and how methylation will affect. And we also just received the 3 wise men brought them to us. The reads from the DNA bisulfite whole genome sequencing from roots from Erodium cicutarium in normal and in drug conditions, but also treated and untreated with azacitidine. So that's all I wanted to share with you. I just wanted to say thanks to my lab mates that have like given me the opportunity to analyze all these data sets that we're producing. I am the guy that does the bioinformatics. My peers are like more in charge of getting the funding, the sampling, greenhouse work. So yeah, without them I would be just some data mining bot, I guess. So thank you, and that's all. Questions?
Thanks a lot, Rubén. That's a lot of, and it was very explained. Like for the one that is doing the gardening, it was very well explained. Questions from the audience?
Okay, thank you. You said that one of the plants is diploid and the other one is tetraploid. Did I understand correctly? In that case, I was wondering, when you look at the methylation, you have multiple variants of the same gene because it's duplicated, or you have 4 copies. Can you quantify if it's the 4 copies that are equally methylated, if it's only one of the variants of the gene? How does this work? I do not really understand how that would work.
Yeah, correct. So in this case, I forgot to mention, we used the, we wanted to make things simple, okay? So we used the, in the differential methylation experiment, we used the reeds that were not treated with bisulfite to locate SNPs that were overlapping with potential methylation sites. So sometimes in the bisulfite methylation, in the bisulfite treated reeds, you can find honest mutations that were there that are mimicking the bisulfite. That means that there's an honest mutation from C to T, all right? If you only analyze the, this position in the bisulfite library, you can uncorrectly assume that that's a mutation and there's honestly an unmethylated cytosine there, when it actually happened is that there's an actual thymine. So we filter those positions with that kind of mutations from the bisulfite. That's a first step. So eventually I'm only analyzing sites that are either, it's like you have 2 alleles, all right? Cytosine, a methylated cytosine and methylated cytosine. And since I have coverage, I don't care about the number of copies of the gene that are overlapping, because I'm going to have a percentage of methylation. It can be 100%, 0%, 50%, okay? So that's the proxy I am using. It is true that I will need to do some more advanced analysis to track down each copy, each allele per separate, but right now we are keeping things simple, and we are just scanning positions and calculating epigenetic structure just with methylation percentage per position.
But in the same line, would the fact of having multiple copies of the same gene facilitate higher ratios or higher methylation, because, well, you have more functional variants of the, more functional copies of the gene, so methylation would be more, well, would be more permissive to methylation, in the sense that would have a lower effect on the expression.
Well, yeah, when there are, I mean, that happens when you have like several copies from this same gene family, I guess you are referring to, or several paralogs, you mean?
Yeah, I meant the multiple copies in the homolog copies, yes.
All right. So not like alleles, you mean like copies,
yeah.
like for example, okay, yeah. So yeah, that depends. It's a birth and death gene scenario. You have several copies, as long as the function of that gene is still like functional. It is redundant, but yeah, the other ones can accumulate mutations. Can, for example, lose methylation context because of the mutation of a cytosine. It can lose that meth epi mutation in that case. So yeah, I haven't explored that in this analysis, but yeah, it could be doable, I think.
Thank
you. Okay, any other?
If I'm not wrong, you have quantified the phenotypic changes resulting from epigenetic changes of a single genome, right? In that experiment.
Yeah, yes, correct.
Okay, so my question is, are you planning to explore whether the phenotypic changes resulting from epigenetic, from the genomes that are highly sensitive to epigenetic changes are higher or lower than the phenotypic changes resulting from genomes that are less sensitive to epigenetic changes?
Okay, so I'm sticking with one species right now. So the question is not yet, but the thing is that I'm more interested in what's the response to a single stressor, but also to extract the component that is regulated by the DNA methylation. Am I answering your question?
What I have in mind is you presented some populations with different individuals,
oh, yeah.
and some of them were more sensitive to epigenetic changes than others, right? So my question is, could you take, for example, the genome that are more sensitive to epigenetic changes and the genome that are the less sensitive to epigenetic changes and replicate the experiment that you have performed by quantifying the phenotypic changes of both genomes?
All right, so this is actually epigenetic divergence. It's not that that this is more sensitive and those are less sensitive. Is this slide what you are referring to?
Yes.
Okay.
So it's not that different points in that graph are more or less sensitive to epigenetic changes?
I wouldn't interpret it like that. It's just that they are different. They have different different percentage of methylation, but it doesn't mean that they are more sensitive or less sensitive to methylation. I think it would be an interesting idea to quantify if there are individuals that are more sensitive, but you can actually get global methylation from them and see if they are in the whole genome have like 25% methylation, 55%, yeah, but that's usually like in a point in their life. It could be variable. So I don't know if we could do exactly that, if there are like genomes that are more sensitive or not. Maybe there are, but definitely I cannot say just looking at this data. Sorry.
Yeah, following with these sort of sources. Yes, my question is whether the amount of variation, phenotypic variation induced by methylation, the structure of methylation in populations, but if this structure you cross with stress, for instance, drought or rivalry, it's possible you can also measure the role or the amount of variance induced by the genotype times environmental induction or source operation, compared with the genotype variation, considering that methylation and different levels, or so different genotypes, for instance. Are you planning to include this factorial design, including stressful, and try to see whether the phenotypic variation may be induced by these genotype times environmental variation?
I mean, that would be interesting to see how different genotypes respond. But right now, the field is pretty rough. For example, this experiment, we use a single parental because we wanted to have control the genetic background. So these are full siblings. So maybe in the future we can add more genetic variation to the experiment and see how each genotype can respond. But right now we are keeping things simple. We probably are going to stick to full siblings. Maybe in the future we can add more genetic variation. Yeah, will be definitely interesting.
Hi, Rubén. Great talk, thanks. I was wondering, so the first part of your talk, you described more of a population level analysis, and there, I think you had like sample size of 36, something like that. So more individuals from different populations, and you have both SNPs and bisulfite sequencing. I was wondering if you had tried like GWAS, like more of a trace association analysis with that seemingly larger data set.
Okay, so I don't have traits for that data set. They are like independent data sets. So the second experiment, yeah, I got the phenotypes and the gene expression. But in the first, I only have the RAD-seq and the bisulfite RAD-seq. So I'm afraid I cannot do that. I mean, it would be ideal to see. We are characterizing, though, soil samples to see if there's some effect, because at least we have the soil in the locations. But yeah.
Cool. And a quick second question. So for this other second part, well, sorry, the first one was also you found some more dense methylation sites in the TEs. So how do you interpret that? Do you in the transposable elements, you have a high proportion of methylation sites. So do you interpret that as mechanisms of like silencing TEs, or how do you interpret that?
All right. So I didn't. What I found, it's not that the TEs were more abundant. It's that within the TEs that I detected, and a specific family, the copia family, is more abundant in the set of differential. All right. So there's not a lot of TEs compared to the total amount of DMRs. Like the numbers here are 20-something, 27, I think, in Erodium cicutarium, 12 or 13 in Erodium Cazorla, compared to the total of, I think, 200 and something in cicutarium, and 40 something in Erodium Cazorla. So there's not, TEs are not enriched in differentially methylated regions. However, if we focus in the TEs, we can see that copia elements are enriched within the TE proportion. So, and that's why I more or less insinuate the conclusion that copia can have an active role in regulation dependent of DNA methylation, because we found that different populations have different levels of methylation in these copial elements.
Any other question? Okay, so we can leave it there. Thanks again, Rubén, for this talk. And yeah, see you next week. No, there is no questions in YouTube. I checked. Yeah. Thanks to all.